Whoa, that's really loud, huh? How's that? Still pretty loud, but I guess better loud than not at all. So, all right, so is that going to be okay? I feel like it's really loud. All right, as long as it's good for you. Yes, thank you for coming to the first class after the midterm, and thank you all for taking the midterm. Every single one of you took the midterm, and so I really appreciate you all staying healthy and strong and doing that. And what that means is during discussion sections this week, the IAs will go over the midterm with you. Before that, I will post a copy of the midterm on Canvas, so you will have it available to go through. They are not graded yet, and the reason for that is I take feedback from the discussion sections to determine how to award potential partial credit for questions that maybe we graded too difficult. So go to your discussion section, argue for the reason why you answered the questions, and we'll take that into account when I do the final grading. Also, the final exam for this class, I know you just took a midterm, you don't want to think about the final, but it's cumulative, and that is because molecular biology builds on itself. So if there's anything you got wrong on the midterm or you still don't understand from the first half, go to your sections, come to office hour, get that clarified. You may very likely see it again on the final exam. Question? Just to make sure, you say go over the discussion sections, like this week's discussion sections, yes. So those of you who have Friday and Monday, those will be your opportunities to go through the midterm, figure out what the answers are, and perhaps provide feedback that will influence our grading, and then the grades will be completed early next week, and you guys will get that. So don't stress over the weekend. Your grade is not going to be posted over the weekend. Enjoy yourself. Have a good Halloween, Dia de los Muertos, whatever you want to celebrate. Any other questions? All right, so moving into new material. So in the first half of the class, we talked about our first two major molecular mechanisms, DNA replication, DNA copied into more DNA, and then transcription of DNA into RNA. Today, what we're going to talk about is how that messenger RNA needs to undergo processing in order to make it a substrate for translation into protein. And we'll talk about the process of translation next week. So as we talked about before, you will have a gene that will have the instructions for transcription. And most of our mammalian protein coding genes are going to be transcribed. In the pre-messenger RNA, the initial transcript that gets made will have often sequences that interrupt the exons. Those are called the introns, and those introns need to get spliced out. We'll also talk about tonight how we need a modification at the 5' end of the messenger RNA. It's called 5' capping to protect that messenger RNA. And then also how we have a modification at the very 3' end, which is called polyadenylation, to add this track of A's, usually about 200 A's onto the end of our human messenger RNAs. And only then, when you have your fully processed, mature messenger RNA, does it go to the cytoplasm to be translated into protein. So tonight, we're going to focus on these three mechanisms. Before we do that, let's see how much we already know about pre-mRNA processing by answering this clicker question. So which of the following do you think are correct about pre-mRNA processing? All right, put your final answers in. Ready? It's got to be more than one of the above, right? Maybe? Yeah? All right, let's go through each one of these. Maybe it's all of the above. Let's see. All right, let's just go through the first one here. So the mature messenger RNA is always shorter than the pre-mRNA from which it came. Is that always true? I'm seeing people shake their heads no. Under what instance would it not be true? Sure, yes.
I was just thinking, like, if the introns being spliced out aren't, like, greater than length of the polyA tail.
Exactly. And there's some messenger RNAs that don't even have introns. And after you make it, you add that 200 nucleotides of polyA tail. So that would not be shorter than the pre-messenger RNA from which it came from. So it could be either or. Sometimes it's shorter, sometimes it's longer. It depends on the actual pre-messenger RNA, how many introns, the size of the introns, the size of the polyA tail that it eventually gets. Does everyone understand why this is not correct? Also has that always statement, which is like in biology is almost never true, but. All right, what about B? Polyadenylation occurs on an exon. Is that true? I see you shaking your head, yes. Yes, you, white shirt. What's your name? Oh, what's your name, white shirt? Isaac? All right, Isaac, why were you shaking your head, yes?
Well, after it gets transcribed, like what's left is the exon.
So splicing takes out the introns. Everything left is the exon. So yes, polyadenylation has to occur on an exon. Otherwise, it'd be removed, right? So remember, these are all parts of exons. The only thing that is not there are the introns that got removed. So yes, polyadenylation happens on an exon in the 3' UTR region of the last exon. Does that make sense? So B is correct. C says transcription continues downstream of the polyA site. Is that true? You want to try? Yeah, good guess. And if you haven't talked about polyadenylation before, that might seem to make sense, but what we'll actually learn about tonight is one of the signals from polyadenylation is downstream of the site where you polyadenylate. And so we will actually talk about that when we talk about polyadenylation. So if this is not clear to you, this one is true. Hopefully it will be by the end of lecture tonight. So C, B, and C were correct. What about D? The length of the polyA tail is encoded by the template strand of the gene. Is that true? Question?
No, it's not true.
Yeah. Yeah, it is added by this polyadenylation machinery that we'll talk about tonight. If this were true, what this would mean is at the end of every gene, you would have 200 T's in the template, right? And that is not correct. That would mean that the template encoded it, but instead we'll learn that there's a special enzyme, polyA polymerase, that can just add a string of A's without needing a template for that. So it was more than one of the above. It was B and C. And if some of this is not clear to you, hopefully by the end of lecture it will be, because we'll be going through all of these points as we go along. Any questions? All right, so we're going to talk about processing of eukaryotic pre-messenger RNAs. First, we're going to talk about what happens at the 5' end of the RNA, and that is a 5' capping addition. Then we'll talk about what happens at the very end, the 3' end, this polyadenylation, this addition of this string of A's. And then finally, we'll talk about splicing, which really boils down to just removing introns and ligating the exons back together. And then we'll review that the enzymes that do these processes actually attach to that CTD of the transcribing polymerase so that these reactions can happen what we call co-transcriptionally. While the polymerase is transcribing and that new RNA is being made, the enzymes are there to already start working on that substrate. All right, so first we're going to talk about 5' end capping. So here is the very 5' end of a pre-messenger RNA. This would be the nucleotide that was encoded by the +1 in the template, +2, and so forth. Why is there a triphosphate group on this very first nucleotide? Yes? So the very first nucleotide that is going to base pair to that template is going to be a nucleotide triphosphate, right? Those are always our substrates. And then the next one that comes in, you're going to make a phosphodiester bond, so that leaves you with just this phosphate here. But there was nothing before this, so it just is your triphosphorylated nucleotide at this position. When capping happens, there are a couple of things. So first, these 2' hydroxyls here on the first 2 bases pick up methyl groups. So you get methylation on those first 2 hydroxyls. And then you get this strange 5' linkage. So there is a nucleotide that gets added so that you have now 3 phosphates together here. And anyone recognize, besides this methyl group, what nucleotide is this? Guanine, exactly, yes. So this is guanine, and there's a methyl group. The methyl group is on the 7 position, so it's methyl 7 guanine. That is the name of the cap that gets put onto this 5' end. All right, your cell's going through all this effort to put these caps on the 5' end of the messenger RNAs. There must be a function for that. And in fact, there's 3 functions of the 5' cap. What do you think one of the functions might be? Put in this funny-looking modification onto the very 5' end of the messenger RNA. What might that do? Yes? So that is one function. So if you think about what you want to do with a messenger RNA, you eventually want it to get out of the nucleus into the cytoplasm, because what do you want to happen in the cytoplasm? Translation. This actually gets recognized by the translation machinery, which we'll talk about next week. So one of the functions is to help export through the nucleus. Another function is to function in translation, and then there's a third one. Any ideas? Yes? Protection, exactly. So nucleases sometimes can very easily see a mono or a triphosphate and start degrading an RNA, but you put this structure on there, and there are not general nucleases that are going to be able to degrade the messenger RNA. Question? So there are proteins that specifically recognize this structure at the 5' end of the RNA, and those proteins help bring it to the nuclear pore complex and ship it out. Same with translation. We'll learn that there are proteins that specifically recognize this structure to help with translation. So this is sort of the shorthand of how we refer to this cap. So it's 5' because it's at the 5' end of the messenger RNA. The M stands for methyl. It's at the 7 position of the guanylate. And the functions are to protect that messenger RNA, help it get out of the nucleus, exported to the cytoplasm, and then be used in translation. So that's 5' end capping. Now let's jump to the 3' end. Okay. So most mature messenger RNAs are going to have 3' poly-A tails. So how do they get that? The way that they get it is there are sequences in the pre-messenger RNA that direct 3' cleavage and polyadenylation. So what do we mean by that? These are the signals that are at the end of that pre-messenger RNA. So there is this poly-A signal, AAUAAA, very specific. And then about 20 to 50 nucleotides downstream of that is where there's going to be cleavage if there is also a GU-rich sequence that's usually 1 to 20 nucleotides downstream of that. What that means is there are proteins that recognize this signal and say, aha, we need to cleave the messenger RNA in the middle of those signals. So can you see now why we need to transcribe downstream of where we're going to cleave and polyadenylate? You need this signal here. All right, so let's look at the mechanism of how this works for 3' cleavage and polyadenylation. So here is the pre-messenger RNA. Here's those two signals right here in the pre-messenger RNA. And there's going to be protein factors that bind to those signals. And don't worry about the names of these factors. You only have to remember the names of two proteins that I've listed over here. But there's a big complex that comes in, recognizes those signals, and one of those is an enzyme that is going to clip the messenger RNA. It's going to cleave at the site, the polyA site that's between the two signals. Once you have cleavage between that site, then you have this enzyme that is nicely named polyA polymerase. Polymerase is synthesizing RNA, and all it's doing is adding that string of A's onto the position that the cleavage occurred. And it ends up being about 200 adenosines. As that A tail grows, there is then a specific protein that will bind to that A-rich sequence called poly-A binding protein. So you'll have multiple poly-A binding proteins, about 15 to 20 nucleotides for each poly-A to bind onto the poly-A tail. So what you end up with then is the mature messenger RNA that has this poly-A tail that happened at the poly-A site. This is where cleavage happened. You'll still have the upstream signal, will still be there in the pre-messenger RNA, but that GU-rich sequence, what happened to that? Got cut off. That is not part of the pre-messenger RNA anymore. All right, so there are 3 functions for the poly-A tail. What do you think those might be? Yes, protection, exactly. Remember that poly-A binding protein is binding up to that poly-A tail? That's going to protect the messenger RNA. What else do you think the poly-A tail might do? Translation, yes. We'll see specifically next week how the poly-A binding protein bound to that poly-A tail actually aids in translation. And one more thing, helps it export, exactly. So very similar to what we saw the 5' end cap, its basic functions. And this is all based on proteins binding to that poly-A tail, helping to protect it, helping get it to the nuclear pore so it can get shipped out to the cytoplasm, and then to participate in translation. Now it turns out that there can be multiple polyadenylation signals in one 3' UTR. And what that can end up is giving us alternative polyadenylation. What that simply means is, in some cases, you will cleave at this second polyadenylation site and end up with a 3' UTR that will be longer than if the cell chooses this first polyadenylation site, and then you end up with a 3' UTR shorter. And so when this happens, usually what this means is the first site might get skipped because maybe it's not quite consensus. Remember, we've talked about consensus before when we talked about transcription factors recognizing DNA sequences. Same idea for RNA. There are RNA binding proteins that recognize these sequences. They deviate a little bit. They might not bind as well. It can get missed, and maybe this is consensus site, so this one will get used much preferably. And then the availability of different RNA binding proteins and the polyadenylation factors in a cell can also dictate how often you do this one versus this one. And this is important because there are sequences in the 3' UTR that are recognized by factors that can regulate the messenger RNA stability as well as how well it can be translated. And we'll talk about a number of those examples in class next week. But for now, appreciate that you can have alternative polyadenylation that gives you 3' UTRs with different sizes, but even more importantly, different sequence that can be recognized by different factors. Does that make sense? All right, so now let's look at how this termination actually happens for the actual polymerase that is transcribing. We said that the polymerase needs to transcribe downstream of the polyase site because it needs to transcribe that GU-rich signal, right? So what happens is when the RNA polymerase is synthesizing and transcribes those polyadenylation signals, the polyadenylation machinery comes in, cleaves. You then get polyadenylation, your mature messenger RNA. But what you have to appreciate is the polymerase is still transcribing down that DNA, down that gene. Excuse me. But where this cleavage has occurred, you now have an unprotected 5' monophosphate on that RNA where the cleavage happened, right? There are nucleases that want to chew up that RNA. Those nuclease complexes will see that unprotected RNA, and they will start chewing up the RNA. So the nucleases will recognize that 5' end of the cleaved RNA, degrade it, and eventually catches up with the RNA polymerase, and it actually causes it to dissociate. So the nuclease activity chewing up that RNA catches up to the polymerase and torpedoes it off of the template. And sometimes this is called the torpedo model because the nuclease, boom, gets the polymerase off of the template. And this is considered to be transcription termination coupled to 3' cleavage and polyadenylation. All right, so any further questions about capping or polyadenylation? All right, let's move on to this final process, which is a bit more complicated and very interesting. So splicing. Splicing is going to be removing these introns and ligating the exons back together. And the way this is accomplished is through a big complex that's generally called the spliceosome. And so what does the spliceosome consist of? Well, you need the substrate. The substrate is going to be that intron-containing pre-messenger RNA. There are then 5 snRNAs, small nuclear RNAs that also participate. So snRNA stands for small nuclear RNAs. And the 5 that participate in general splicing are U1, U2, U4, U5, U6. What happened to U3? It does not participate in messenger RNA splicing. It actually participates in ribosomal RNA maturation. They are called U's with a number because they are U-rich RNAs. There's a lot of U's in them, and they were numbered based on when they were discovered, not necessarily what order they do things. In addition to these snRNAs, and we'll see why the spliceosome uses these small nuclear RNAs in a few slides, there's also over 100 protein factors that are part of the spliceosome to accomplish this very important task of removing precisely the intron. Think about it. If it's off by one nucleotide, if it leaves one nucleotide of the intron in or removes one nucleotide of an exon, what happens to that messenger RNA? Frameshift, right. And if you don't know what a frameshift is, we will go over that next week when we talk about translation. But if you have the wrong nucleotides left in your mature messenger RNA, you're going to change what it encodes for. You're going to change the protein coding capacity. So this machinery needs to be very precise to get it right every time, or if it doesn't get it right, get rid of that messenger RNA. And we'll talk about how that occurs in a couple of classes. So splicing essentially is 2 transesterification reactions. What transesterification means, you're just exchanging phosphodiester bonds. Remember, it's phosphodiester bonds that are linking together your nucleotides. And so you're going to have 2 transesterification reactions each time you remove an intron and then ligate together the 2 exons. So let's look at the chemistry of how this occurs. So when you have your pre-messenger RNA with your intron, within that intron, there is going to be what's called the branch point A. It's always going to be an adenosine nucleotide, and its 2' hydroxyl is going to be available to attack the phosphodiester bond that is connecting the last nucleotide of the exon to the first nucleotide of the intron. When this attack occurs, this is the first transesterification reaction. It forms a new phosphodiester bond between this 2' position in the branch point A and this phosphate here. And what does that do? It releases this exon. You now have a hydroxyl at this exon, which can then attack the phosphodiester bond that is connecting the last nucleotide in the intron to the first nucleotide in the next exon. When that attack occurs, which is your second transesterification reaction, it releases this intron and then ligates together those two exons. And you'll be left with this intron that has this sort of funny structure. Remember, you had this branch point A using its 2' hydroxyl to bond to this phosphodiester bond. This is often called an intron lariat. Have you heard of the term lariat? Like cowboys, the ropes they use to catch cattle sort of looks like this. That's where the name comes from. This will get degraded. This will be part of your mature messenger RNA. All right, so let's look at how the spliceosome is able to precisely find where are the introns, where are the exons, where does these transesterification reactions need to occur. So there are 4 general signals that are going to mark where splicing needs to occur. There is the 5' splice site. So this is where that first attack is going to happen. And you'll notice that, so what we're showing you here are the consensus sequences. So these numbers mean that 100% of the time there is a G and then a U in the first 2 nucleotides of the intron, and then you have a little bit of flexibility around that. There's also going to be then the branch point A. You're 100% of the time going to have an A there that is going to do the attack. And then there's also a high preference for nucleotides surrounding that A. There is a polypyrimidine tract, so a stretch of 10 to 12 bases that are pyrimidines. Which ones are our pyrimidines in RNA? C and U, very good. And then finally, you're going to have your 3' splice site. And again, these last 2 nucleotides in your intron are usually pretty invariable. About 100% of the time, you have an A and G there, and that will mark your 3' splice site. So these are the 4 different signals that are going to help position the spliceosome to know where to remove the intron and ligate the exons back together. But how does the spliceosome actually recognize these positions, especially these positions with these very high consensus sequences? Does anybody have a guess as to how these sequences might be recognized by the spliceosome? Thinking about the components that are in the spliceosome that we talked about. Any ideas? So we talked about 2 major components that are in the spliceosome besides the pre-messenger RNA. So it's got to be one of those 2, right? Is it the proteins or the snRNAs? The snRNAs might be able to base pair. Exactly. That is what happens. So the U1 snRNA has a sequence at its 5' end that can base pair to those sequences that are at the 5' splice site. U2 snRNA has sequences within it that base pair to the sequences surrounding the branch point A. Let me just make sure that we all understand that RNA, even though we often think of it as being single-stranded, can actually base pair. It can base pair to itself. It can fold back on itself. We call that intramolecular base pairing. We also can have RNAs base paired to other RNAs, such as this stretch in U2 snRNA that is base pairing to this region at the branch point A. We call that an intermolecular base pairing event. Notice that this A is actually not base paired. That branch point A, the one that's going to do the chemistry, is bulged out. It does not base pair to U2 snRNA, and that's very important to position it so it can do that attack at the 5' splice site. Sometimes when you see RNAs that fold back on the cells, we sometimes call that a stem loop. There's a little bit of stem, and then there's a loop up here. Sometimes we call it a hairpin, you know, like those bobby pins you can put in your hair. So if you hear those terms, that's what it's referring to. Okay, so the U1 snRNA base pairs to the 5' splice site. The U2 snRNA base pairs to the branch point A. How do we know that? How do we know that these snRNAs need to base pair to these sites for splicing to occur? Well, it's like anything that we know in science. Experiments led us to this conclusion. And so one of the important experiments that was done to test this idea that U1 needs to base pair to the 5' splice site is that researchers made a little model gene that had an intron for splicing, and then they mutated one of those highly conserved sites that's near the 5' splice site. So they changed that G to an A. A does not base pair to C. Put that in the cell, and lo and behold, they got no splicing. So from that, you could conclude either you need a G at that position for splicing to occur, or you need base pairing to occur, and it doesn't really matter what the nucleotide is. So to test between those two models, what do you think the next experiment is that they did? If you want to test, is just base pairing important here? What could you do? Yes? Make something that will base pair to the A. Change your U1 snRNA. Absolutely, that's what they did. So then they also put into the cell a mutant U1 snRNA where they changed the sequence in the U1 so that it could reestablish base pairing to that mutant pre-messenger RNA, and that completely restored splicing. So from that, you can conclude it's not important that it's a G here. It's important that you are able to base pair to stably recruit the U1 snRNA and allow splicing to occur. We call this a compensatory mutation. So we are compensating for one mutation with a second mutation. Does that make sense? All right, now let's go through the basic steps of how the spliceosome recognizes these signals and carries out the splicing reaction. So the first thing that's going to happen in your pre-messenger RNA is that U1 is going to recognize, and so when I say U1 or U2 SNRP instead of snRNA, what that means it's the snRNA with all the proteins that it's associated are coming in as a complex. But it is the RNA part that is base pairing to that 5' splice site, and then it's just bringing all its proteins with it. U2, SNRP, the snRNA plus its proteins, is going to recognize that branch point A. Once those two complexes join, then U4, U5, and U6 can join the complex. There is a rearrangement that then causes U1 and U4 to leave, but importantly brings that branch point A into the vicinity of that 5' splice site so that you can get that first transesterification reaction. So that branch point A is forming that 2', 5' bond with the 5' end of the intron. That then leaves this free 3' hydroxyl at the end of the first exon that is then able to carry out this second transesterification reaction where it will attack the phosphodiester bond, linking the end of the intron to the beginning of the next exon. That will result in these two exons now being joined together. We'll have the release of that intron lariat. There's then an enzyme that debranches, so breaks this funny 2', 5' bond. You now have a linear RNA with no protection at the 5' end, no protection at the 3' or 5' end. It gets rapidly degraded. All right, so there's a problem with especially human messenger RNAs is that they are huge. They're quite long, and the reason why they're so long is not because of the exons, it's these introns. We have huge amounts of sequence interrupting our exons. So on average, exons are only about 150 nucleotides, and they're interrupted by introns that can be thousands of nucleotides long. So this makes it very difficult for the spliceosome to find those right splice sequences. And one of the ways that they are helped is by what we call exon definition, meaning that there are proteins that bind to sequences within the exons that then make protein-protein contacts with the spliceosomal machinery to say, hey, here I am, I'm the exon, so use this 3' splice site and this 5' splice site. The spliceosome is still recognizing the signals it needs to see, but now instead of searching throughout these thousands and thousands of nucleotides, it's helped by being brought close to the exon through proteins that recognize sequences within the exon. These RNA binding proteins, there's a whole class of them, but in general, they're often referred to as SR proteins because of the amino acids that they are rich in. S stands for serine, R stands for arginine. So they are just rich in serine and arginine, and that's why they're called SR proteins. There's dozens of different SR proteins. And what they do is they bind to sequences within the exon that we call exon splice enhancers. They are enhancing the recognition of that exon so that the spliceosome can get rid of the introns that are flanking it and then ligate those exons back together. All right, so now we've talked about a number of different signals within the pre-messenger RNA that are going to be important for accurate and efficient splicing. And so who can name one of the intronic signals that, if it were mutated, you would definitely affect splicing? Sure. The branch point A, yeah. If you don't have that branch point A, you're not even going to have the first transesterification reaction. So yeah, you don't want to mutate your branch point A. What else would be a problem? Yes? The polypyrimidine tract. Yep, that's one of the signals that we need. What else do we need? Two other signals. Sure. Were you raising your hand? No? Okay. Yes? You need a what? The enhancers are going to be very important for the exonic signals. So those exon splice enhancers we just talked about. Good. What about, we have two more in the intron. Yes, Maddie? Yep, the 3' and 5' splice sites. So those four signals that we initially talked about, the 5' splice site, the branch point A, the polypyrimidine tract, 3' splice site, you get a mutation in any one of those, it is going to interfere with splicing at that position. We then also talked about the exons having sequences within them that need to be bound by these SR proteins to help define them. We call them exon splice enhancers. It turns out there's also such a thing as exon splice silencers. What do you think they would do to the exon if a protein binds to a silencer? It can cause skipping of that exon. So, and that's important. Sometimes we do want to skip exons. We'll talk about alternative splicing in a moment. So, the exon splice enhancers will make that exon be included in the mature messenger RNA. Silencers will cause it to be skipped, to be silenced. There can also be sequences like this in introns that will recruit specific RNA binding proteins that will cause nearby exons to be used or to be skipped. Now, the recognition that these sequences are important for splicing, especially that there are actually sequences in exons themselves that influence splicing, was very important for figuring out what had been a mystery for a really long time, these silent mutations. So what do we mean by that? So many of you probably know that for many of our amino acids, there are codons that can be different in sequence but encode for the same amino acid. There's some redundancy. So UUC encodes for phenylalanine, but so does UUU. Both encode for phenylalanine. So if you change a C to a U in an RNA, it shouldn't have a mutation, right? It shouldn't have an effect. You should still encode for the same amino acid. But what we were seeing is sometimes that really did affect gene expression. And so why would that be affecting gene expression? Well, what we now appreciate was disrupting a splice enhancer or potentially creating a splice silencer. By changing that nucleotide, you could change how those SR proteins might actually be able to bind to that specific sequence. So the change would still encode for the same amino acid, but if it changes the efficiency of splicing, you might end up with only half as much mature messenger RNA to make the protein that has that amino acid sequence. Does that make sense? So this is actually a really good example of a silent mutation that has disease relevance. Comes in the case of spinal muscular atrophy, or SMA. Some of you probably have heard of this really horrible disease. It's actually one of the most prevalent genetic diseases that causes death in infants. The muscles just atrophy and waste away. You can't breathe. And initially, patients who would have this would not live very far through after being born. And the reason is there is a protein called SMN, survival motor neuron, and that is its job. It is to help the motor neuron survive so you don't get this muscular atrophy. And it turns out that in our human genome on chromosome 5, we actually have 2 copies of SMN, and they both encode the exact same protein. So you would think that if you lost one of these, SMN1, you still have SMN2. You should be able to make this protein and survive. Patients who have SMA actually are missing SMN1, and they do not make sufficient SMN protein product. And the reason is because there is a single nucleotide change in exon 7 that changes this C to a T. And this affects splicing. So in the SMN2 gene, initially it was thought that this change was disrupting an exon splice enhancer. Very recent research over the past few years figured that it actually doesn't disrupt an enhancer, but it creates a new silencer. The effect is essentially the same. It causes exon 7 to be skipped 70% of the time. If you have a mature messenger RNA that is missing exon 7, you do not make functional SMN protein. However, if you have SMN1, you're in great shape because it does not have that sequence change. Splicing works great. You get all of the exons you need, and you make plenty of SMN protein. The problem comes in is if you have any kind of mutation that knocks out expression of SMN1, then your only choice is to make from SMN2. And about 30% of the time it does include exon 7, but this just is not a sufficient amount of SMN protein to survive long beyond birth. And so this splicing change, this change that simply makes a splice silencer, is the basis of this disease SMA. So now that we know the molecular basis of this horrible disease, how can we cure SMA? Can we? What would you do? You know what the problem is. You know what your options are. What could you do? Any ideas? You don't have enough of this protein product. Yes, Lydia. Introduce a gene to make more of that protein, right? So a gene therapy approach. In fact, that is one example. What about if you know there's a splicing problem? Like if you could just fix the splicing of this, right? Yeah, so that's actually the basis of 2 other therapies. So there's actually 3 therapies that have been developed over about the past 10 years. The very first one came with once it was understood that all we need to do is get the cell to include exon 7 more often. And it turns out that just by adding an oligonucleotide, so a little stretch of modified RNA, into these patients, somehow it gets into the cell, reaches the pre-messenger RNA, and it kind of blocks the proteins that do the silencing, that like stop that exon from being included. And now you go from it's 30% being included to often well over 50, 70, 80% included, and then you make enough of this SMN protein. So you've changed the splicing of this gene they already have. Another therapy came out several years ago that does exactly what Lydia suggested. Well, let's just add the gene back. And so we'll talk a little bit more about gene therapy when we talk about viruses, because gene therapy often uses viruses to introduce a human gene that is missing. In this case, what the company did is they stripped the virus of all its coding sequences that could cause harm, put the human gene in for SMN1, let's make the one that's spliced really well, and then target it to the neuronal cells that need to receive this. And this, because it's a virus that has the gene in it, once it gets into the cells, it can always make it. This is a single injection that lasts their entire life. You may have heard of this one because it made a lot of news when it came out because it was a $2 million therapy. And, but it's just one dose, and then there's no treatment the rest of their time. So it is essentially a cure. Another one that came out that's much more affordable and has the same idea of correcting the splicing actually doesn't use an RNA oligonucleotide, instead uses a small molecule, a little drug that people take it orally. And they still don't even know the mechanism of how it corrects the splicing, but somehow it does. And all three of these have now been approved by the FDA and are actually having really remarkable results in real human patients. And I think, you know, all of these success stories are really exciting and really emphasizes how important it is to know the molecular mechanism of a disease, and then you can start to think about therapies that can actually work in real human patients. All right. So just to review again, when we are talking about these various enzyme complexes that are involved in putting the cap structure onto the RNA, putting the 3' poly A onto the RNA, splicing, these enzymes can all associate with that C-terminal domain of that polymerase as it's transcribing. What that means is as the new messenger RNA is being synthesized, the enzymes are already there to immediately put a cap on, start protecting that messenger RNA, start splicing the RNA while it's still being synthesized. And then finally, we already talked about that co-transcriptional polyadenylation, cleavage, and termination mechanism. We also said when we talked about that CTD, there's a lot of amino acids that can be potentially phosphorylated. Do you think these processing factors are associating with the CTD when it is in the phosphorylated state or the unphosphorylated state? Phosphorylated, right, because when transcription is just starting, when we are bringing the pre-initiation complex to the promoter, it is largely unphosphorylated. And it's only once you get TF2H that has that kinase activity there and you start transcribing, you put phosphate groups on there, and that's what recruits these enzymes to sort of bind along this scaffold and be ready to start working on that pre-messenger RNA substrate. Any questions about this? All right, so now let's talk about regulation of pre-messenger RNA processing. So we talked before about how we could have alternative polyadenylation, so we can choose a different cleavage point on our 3' UTR, was our alternative polyadenylation. Now we're going to talk about alternative splicing. We'll define what we mean by alternative splicing. And then we'll talk about some examples so you can see how splicing repressors and activators can work to determine which exons are going to be included or skipped during processing of a given messenger RNA. So when we talk about alternative splicing, what we mean is we will have initially our pre-messenger RNA that will have all the sequence that was encoded by the gene that will be transcribed. And in some cases, you might have an isoform where 1, 2, 3, 4, all 4 exons are chosen and ligated together. These dashed lines represent the introns, or they're being spliced out. Other times, we might skip one of these exons and just have exon 1, 2, 4 come together to make another isoform. Those 2 different mature messenger RNAs will have different coding potential, right? There's different sequences that are included or excluded. You can therefore make a different sequence protein by this mechanism of alternative splicing. Does everyone understand what I mean by alternative splicing? So what can happen in a cell? You might have one cell that only makes this isoform, another cell that maybe only makes this isoform. You might have another cell that 50% of the time it makes this, 50% of the time it makes this, another cell type maybe 80, 20. You can have different ratios. And so what's going to determine that? What's going to determine the different splicing patterns that you're going to get for the same pre-messenger RNA within a cell? The proteins that are recognizing the enhancers and the silencers, exactly. The sequence is the same in every cell. The proteins that are available, the RNA binding proteins that are going to recognize those sequences, their levels can be very different in different cells. And that will determine, are they actually being bound and recognized or not? Does everyone get that? All right. Now we're going to go through a very famous example of alternative splicing that actually regulates the sexual differentiation of fruit fly Drosophila. And this is really just to show you an example of how the same messenger RNA can be differently recognized and alternatively spliced. So fruit flies, you have ones that are XY that are male and XX that are female. And one of the ways this differentiation process starts is through alternative splicing of a gene called sex lethal. So if I tell you that sex lethal is only expressed in females and not males, what do you think that name comes from, sex lethal? Which one is it lethal for if you don't have it? Females, exactly. The females make it, males don't. So if you're male and you delete that gene, it doesn't matter. But females need it. So it's called sex lethal because it's only lethal for the XX flies. And the way this gets set up is very early in development, because the females are XX and the males are XY, a version of the sex lethal protein is made only in the females. Later in development, the sex lethal pre-messenger RNA is made in both males and females. So you get the same exact pre-messenger RNA, but keep in mind only the females have made a version of sex lethal protein earlier. So that protein is available only in females to bind to a sequence in the intron between exons 2 and 3, close to exon 3. And what does it do? It actually hides exon 3. It causes skipping of exon 3. So in females only, you get a mature messenger RNA that has exons 2 and 4 ligated together, and then there's other exons on either side. This makes sex lethal protein, which goes back and binds more of its pre-messenger RNA. In males, there's no sex lethal protein to begin with. There's nothing to bind and hide exon 3. So the spliced mature messenger RNA has exon 3. And it turns out that exon 3 has a premature stop codon in it. So the translation machinery will not make it through the protein. And in fact, this messenger RNA becomes destable. And you do not get any functional sex lethal protein. All right. Excuse me. So this is what we just went through here, how sex lethal protein regulates its own splicing. How does it regulate its own splicing? It causes exon skipping. Does everybody see that? So it works on its own messenger RNA that way, but it also sees a sequence in a gene called transformer, or TRA for short. This sequence is between exons 1 and 2, and what does it do? It again hides that exon that it's bound next to, causes skipping of exon 2. So now when TRA, pre-messenger RNA, gets spliced, in females, which are the only ones that have sex-lethal protein, you skip exon 2, you ligate 1 and 3 together, and that makes transformer protein. Males don't have sex-lethal, they can't skip exon 2, they make a pre-messenger RNA then that has exon 2. Again, this exon also has a premature stop codon. It can't make the functional protein. So only females then end up having transformer protein. What does transformer protein do? Turns out that transformer protein forms a complex with 2 SR proteins that are common in males and females and many different cell types, this RBP1 and transformer 2. When TRA protein is available, it recruits these SR proteins to exon 4 in a pre-messenger RNA for a gene called double sex, DSX. When TRA, along with its partners, binds to exon 4, it actually causes inclusion of that exon. Inclusion of that exon results in a mature messenger RNA that has exon 3 ligated to 4, and it turns out that exon 4 has its own polyadenylation site. So this then makes a female version of the double sex protein. Males don't have TRA. They can't recruit these general SR proteins. Exon 4 is not seen as an exon. It is skipped in the males. So now males make a double sex protein from a mature messenger RNA that is missing exon 4 and instead has different exons. These 2 different versions of the double sex protein are actually transcriptional repressors. The female version represses male genes. The male version represses female genes. And so this cascade of alternative splicing is what sets up a lot of the sexual differentiation that happens in these fruit flies. So in answer to this question, how does TRA protein regulate splicing? How would you answer that? It promotes alternative splicing in a way. What actually is it doing when it binds? Inclusion. It's promoting inclusion of exon 4. Exactly. All right, so why did we go through all this alternative splicing in Drosophila? The point here is not to memorize the names of these Drosophila genes, but just to show you examples how specific RNA binding proteins can bind to sequences in a pre-messenger RNA and cause either exon skipping or exon inclusion. And so there are many, many RNA binding proteins that will recognize specific sequences in exons or in introns that will affect splicing, that will give you the result of alternative splicing. So alternative mature messenger RNAs that then can encode for different proteins. Does that make sense? And here is one of the most extreme examples of alternative splicing, just to give you an idea of how complex this can be. So this is the alternative splicing of a gene called DSCAM. DSCAM stands for Down Syndrome Cell Adhesion Molecule. It encodes neuronal proteins that are needed for synaptic connections. It was first described in Drosophila. Humans have this DSCAM gene also, and it is just as complicated, the splicing of it. What happens with this gene when it is transcribed into this very long pre-messenger RNA? These 3 black lines here, these are what we call constitutive exons, meaning Every D-scan protein is going to have these first 3 exons included. But then it can choose one of 12 of these blue exons for the next one, then one of 48 from these next, and then it'll include these 2 always, and then one of 33, and then all these additional exons down here. If you do the math for every combination that can possibly occur here, there's over 38,000 possible isoforms of mature messenger RNAs you can make from this. Each one can encode a different type of protein that has different properties for making synaptic connections. And so what this means is when we think about our own human genome, well, we only have about 20,000 protein coding genes. Each protein coding gene is potentially alternatively spliced into multiple, dozens, hundreds, sometimes thousands of different isoforms. And so we don't really even know all the complexity of all the different proteins we are capable of producing. Does that make sense? All right, let's do a clicker question. All right. So above is the structure of gene X with the indicated nucleotide positions. And next to it is a northern blot showing the detection of gene X mRNA in 4 different cell types using a probe complementary to exon 2. And then note that the mature mRNA gets a 200 nucleotide poly A tail. The results support which of the following possibilities? Take a look at all this because it might disappear when I put this guy. Okay. Okay, yeah. There's nothing hiding under this right here. Ten more seconds. Final answers. All right, so tight competition between C or E. Let's go through it. Who wants to offer their answer and reasoning? Yes, Madeline.
I said C, even though there's no band next to the markers, we can see that the controls still work because the reciprocal RNA is present.
So C says exon 2 is skipped during splicing of gene X in the C cells. All right, so if exon 2 is skipped, what would we see on our northern blot? Nothing, exactly. If you don't have the sequence for the probe to bind to, you're not going to see anything. The RNA will still be there, but you're not going to detect it. We look over here at C, and just as Madeline said, first we check our control. RNA looks good. Our control is there. RNA is good quality, but we detect nothing, and that's exactly what we would predict if we put our probe in exon 2 and exon 2 got skipped out. So that would make sense for the result we see in C. So C is correct. Let's go through the rest of these and see if maybe there are more than one of the above. A says gene X mRNA is mature and fully spliced in the A cells. What would we expect if the mRNA is mature and fully spliced? Yes?
The removal of the intron.
So removal of the intron. So what size would that? So we know our pre-messenger RNA is 450. If we remove the introns, what are we down to? 350, right? Because each one is 50 nucleotides. So 450 minus 100 is 350. Fully polyadenylated is because I say it's mature. So that would give us 550. All right, is that what we see in the A cells? No. What happened in the A cells? Completely pre-messenger RNA and got the poly A tail, right? 650. That would make sense for that. Does everyone see how we're doing this? So A is not correct. B says intron 1 is retained during splicing of gene X in the D cells. So we just said if we have it fully spliced and polyadenylated, we would be 550. If we retain intron 1, what size would we be? 600. Is that what we see in the D cells? No. What actually happened in the D cells? Introns removed, fully spliced. It's mature. When we say mature, it means it got polyadenylated. So B is not correct. We already know C is correct. Uh-oh, we better hope this last one's correct, right? Gene X does not undergo splicing in the C cells. So we already said what would happen if it didn't undergo splicing? Be 650. We look in the C cells. We should see it, right? Because if it didn't undergo splicing, it still has exon 2, should be detected by the probe. We don't see anything, and it's more likely that either it didn't get transcribed, that'd be another reason we wouldn't see it, or we designed our probe over an exon that happened to get skipped in that cell type. So the only correct answer was C. Does everybody see where we get that? And then this is just going through the simple math that we talked about when we were predicting the various sizes you would get through all of this. And this will be included with the slides or the clicker questions when I still have one more slide because many of you wanted to know, how do you get the RNA out to the cytoplasm once it is mature? So once you have your RNA that is 5' cap, 3' polyadenylated, and has had its introns removed, spliced correctly, there are proteins that bind to that mature messenger RNA. We talked about the poly A binding proteins binding to the poly A tail. There are cap binding complex proteins that bind to that 5' cap, and a whole bunch of other types of proteins that we're just all going to generally call exporter proteins, because what they do is they recruit the RNA to the nuclear pore complex. So this is this big protein complex that goes through that double membrane of the nucleus. The proteins that are bound to the pre-messenger RNA interact with the nuclear pore complex proteins, help transport the RNA into the cytoplasm. Once in the cytoplasm, those exporter proteins leave the messenger RNA. New RNAs, including those that are going to help with translation, bind the messenger RNA, and the exporter proteins go back in the nucleus to help bring out subsequent messenger RNAs. Any questions about that? All right, now we are done. So have a great weekend. Have a good Halloween.